Metabolism and enzymes. Metabolism is the breaking down and building up of molecules. We've seen it before. Condensation reactions are actually anabolic, which means that molecules are being built from smaller molecules. So we have the monomers being put together by condensation synthesis into a polymer. Metabolic reactions can be anabolic or catabolic. Hydrolysis reactions are catabolic. In hydrolysis reactions, a polymer is broken down into its monomers. This is a catabolic reaction. The total of all anabolic and catabolic reactions in a living thing is called its metabolism. There are a lot of reactions going on. Metabolism is all the chemical reactions occurring constantly in a cell that maintain homeostasis. It is the orderly, stepwise series of chemical reactions from the initial reactants to the final products. Each step within a pathway requires a specific enzyme. Metabolic pathways exist because many intermediate steps are needed to produce complex molecules. So you can't go straight from compound A to compound G. There are a lot of intermediate steps, as we saw in cellular respiration. It's not possible in biological systems to have a single reaction that could produce complex molecules from simple reactants. For example, cellular respiration could never occur in just one step as it's shown here. Instead, it's actually a series of many small steps. And if you look very closely, you'll be able to pick out here our starting point glucose and glycolysis and then the Krebs cycle. And you can see from this that other molecules besides glucose can enter this pathway at a number of different locations. So glycogen can be broken down into glucose, it's true, but fats can also enter partway through glycolysis, as can other sugars. Chitin can be broken down into fructose and enter glycolysis that way. And you can also have different amino acids entering the pathway at Krebs cycle in a number of different locations. We know that anaerobic respiration can take place and lactate can be produced, but we know that that can be converted back to pyruvic acid and then enter the Krebs cycle as well. Each one of these reactions, as we convert one reactant to another product, requires a different enzyme. And all the reactions taking place in the living thing put together are its metabolism. So cellular respiration is part of metabolism. And as we can see, one pathway can lead to several others. Intermediate products in one pathway can be starting reactants of another pathway. So once again, for example, pyruvate can actually be converted into much smaller products and a lot of ATP in the Krebs cycle, but can also be converted to lactate or ethanol depending upon which organism it's in. Many steps mean many places to control the overall reaction, and this happens through the availability of enzymes, and sometimes the products themselves are the limiting factors to the reactions. We'll see how that works. Let's take a look at some characteristics of metabolic pathways. One characteristic is that the relative concentration of reactants and products really makes a difference. It matters. The concentration of the reactants and end products determines the net direction, forward or backward, of our metabolic reactions. In this example, we can see what goes on inside of a plant. Plants do a lot of photosynthesis, and as they do so, they create a lot of glucose. The glucose can't all be stored as glucose, so it gets converted by reaction involving the enzyme maltase into maltose, and then involving amylase into starch. And that starch can be stored, for example, in potato tubers. Now, because the starch is whisked away as it's formed, it's never really allowed to accumulate where the glucose is being produced, that is, in the leaves. It's produced there, but then it's moved down to the roots of the potato. And as a result, the starch never accumulates and the reaction never runs in reverse. Now, this might actually happen in a test tube, but in reality, in living systems, it doesn't happen because as soon as products are formed, they move elsewhere. This drives the reaction in one direction. It keeps it going in this direction so that any new glucose that's produced gets converted into starch. It would be counterproductive if the starch was converted back to glucose, and that's avoided because the starch is never allowed to build up because it's moved on to a different location. In our bodies, it's another story. When we consume the starch, it goes into our digestive system and ends up in our small intestines. And the starch actually never really accumulates. Even if you continue eating starchy bite after starchy bite after starchy bite, the starch will get broken down by amylose into maltose and then by maltase into glucose. But the glucose never is allowed to accumulate inside the small intestine because as it's produced, it gets absorbed by the walls of the small intestine and enters the circulatory system and moves away into the liver. So the amount of glucose is always in short supply, and because more starch is coming in, the amount of starch inside the small intestines is always in high supply. So this drives the reaction in this direction. So relative quantities control which direction the reaction actually goes, whether it goes from left to right or right to left. At equilibrium, the rate of reaction in the forward direction equals the rate of reaction in the reverse direction. And as we just saw in the examples, we don't generally reach this point in living systems because this presupposes sort of a closed system within a test tube, let's say, where A gets converted to B, B gets converted to C, but then some of C is going to get converted back to B, and that will also get converted back to A. And it's just based on the number of collisions with the enzyme. So at some point you reach equilibrium and you'll have a steady quantity of A, B, and C within the test tube. That's not so within the examples we just looked at, where the reactions are being driven in a direction because as the product gets produced, it no longer remains in the same location. The second important idea is that enzymes are essential. Enzymes, organic catalysts made of proteins, speed up the rate of reaction by lowering activation energy. Activation energy is the energy needed to trigger the formation of new bonds. In this graphic, we can see that the progress of a reaction without an enzyme requires a huge input of energy, maybe in the form of heat. In the human body, it's just not practical to keep our operating temperature very high because at certain temperatures, not too far above normal body temperature of 37 degrees Celsius, our proteins in our body, many of which are enzymes, will begin to denature, that is, change shape and therefore become non-functional. So there has to be another solution. In living things, we use organic catalysts, and you can see that the organic catalyst actually lowers the amount of energy necessary to get the substrate to go through a reaction to produce the product. Substrate and product are the same in either case. It's just that with an enzyme makes this reaction a lot more likely to happen in the absence of added heat energy. And this is completely what we want happening inside of a living thing. This is why we have so many enzymes catalyzing our reactions. So the way this might work is that if you have glucose and maltose is to be built inside of a living thing, you would need the enzyme maltase, and maltase would catalyze that reaction to produce maltose and therefore would require less energy for that reaction to go. Now, glucose will actually react with another glucose to produce maltose in the absence of maltase and in the absence of any added heat energy, but this takes a long time and way too long for this reaction to be of any use to a living thing, and so we do need this enzyme. And of course the same reaction can occur in reverse. Now these enzymes don't actually participate in the reaction, and they're not changed, and they can be used over and over again. So they don't change the nature of the reaction. Glucose will still react with glucose to produce maltose in the absence of maltase, but the thing is, once the maltase has finished catalyzing that reaction, it can go on and catalyze another reaction between other glucose molecules to produce maltose, and so on. And this can happen many times very quickly over and over again. Okay, so that's a brief overview of the characteristics of metabolic processes. In the next video, we'll take a look at how enzymes work.